I'm Bob Dustman. I'm the director of the McGovern Institute for Brain Research at MIT. The McGovern Institute is a brain research institute with a dual mission: understand the brain, and then apply that knowledge to help people suffering from many forms of brain disorders.
Conditions ranging from dyslexia and autism in children to Parkinson's disease, Alzheimer's disease, macular degeneration in the elderly, as well as depression and schizophrenia. All of that work depends on the strong foundation in basic research. We currently have 16 distinguished faculty members, including 1 Nobel laureate and 5 members of the National Academy of Sciences. We attract people who want to be part of this team, who want to work on big problems that can't be solved by one person working in their lab. We put all of these people together in the same place so we can find these links, find the common threads that tie together these basic brain mechanisms that contribute to so many different aspects of human cognition. We believe that basic research spanning molecules to mind will lead to the breakthroughs that will benefit society in the future. This belief is shared by the McGoverns themselves.
My wife and I said, now we have the tools to really tackle how to understand the human brain and help to solve mental illness and brain disease. We have at the McGovern Institute people from cognitive and brain science department, people from the biology department who know all about cellular and subcellular work and the neurons, people from electrical engineering who run the supercomputers to help us do the brain scan analysis. We have people from mechanical engineering who make nanotubes so we can insert into the brain.
I think that's the genius of the Institute. It's the aspect of total collaboration, bringing many disciplines together. We have created and imbued a sense of entrepreneurial research.
We can start research for which no other source of funding is available.
From the first aha idea of a scientist at the lab bench,
some of them are going to turn into be a major discovery. We're going to get a cure for a major mental illness or brain disease.
I'm not sure if my reputation was earned or created by me, but I had a very strong sense of being the most normal person on television. I was diagnosed on a Monday. I knew that bipolar was once more commonly called manic depression. I was preparing my first speech, which would happen to be at the McGovern Institute for Brain Research.
And this is the most...
I joked at the time that MIT stood for my mental illness talk. Made a Nobel laureate laugh. Every time I come here, I find the stories so inspiring. Perhaps the best way to get a sense of the McGovern Institute is to hear just a few of these stories.
I'm Nancy Kanwisher, and I'm an investigator at the McGovern Institute for Brain Research. Until recently, it's been very difficult to study the neural basis of cognition because this was sort of thought to happen all over the brain in a kind of disorganized way. It was very controversial whether high-level aspects of cognition, thinking, are organized into compartmentalized separate bits.
So what we're expecting to see is a little part of your brain right in there that responds when you look at faces. Like right now, it's going. It just turned off. Now it's on. Now it's off.
More like a Swiss Army knife with a set of highly specialized components. Years ago, I had a really brilliant graduate student named Rebecca Sachs who said that she wanted to look for brain regions that are involved in how we think about what another person is thinking. And I thought that was a pretty cool question, but I just didn't believe for a moment that the brain had specialized regions for things that are that abstract. And boy, was I wrong. The region of the brain that's selectively involved in thinking about what another person is thinking is a prime candidate for a region that would be affected in autism. As best as anybody can tell, the core deficit in autism is one of being able to understand other people's thoughts.
Nancy's the perfect example of someone who spent her whole life understanding human cognition and in recent years applying brain imaging technology to understanding human cognition.
You ready?
And now she's going to take a battery of these tests to get that very basic understanding of what is it that's going wrong in the brains of these kids suffering from autism.
Hi. I think when Jackson was about 2, when most parents are expecting their children to start making sounds and say words, he said a couple things. He said kitty and he said daddy. He never actually lost any language, but he just didn't gain any more words than that. I think those signs were enough as an educator for me to know that there was something up. You hear things about, children who are on the autism spectrum who just love street signs or just love music. He loves everything.
I love books. They are really fun to read. I've recently read Diary of a Wimpy Kid book 3.
When we walked into the McGovern Institute, he was just flabbergasted. I think he wanted to go out and come back in again because he just didn't know what to think about it. And then he just got comfortable there very quickly. What was nice about the passport for Jackson was he could have something tangible.
This is when I got my brain scan and there's a picture.
Something to carry around with him all day.
It's an x-ray view it's kind of like an x-ray view into my body when I hold it up like this.
Scanning any kids is tricky. It's right at the edge of what you can do in neuroscience research.
They're looking for keys of autism inside my brain. It's pretty freaky.
I tell everybody that we are at MIT doing this because the finger is on the pulse there. The fact that we could go in and get a picture of his brain, I mean, where else can you do that?
Do you think what you're doing is going to help some other kids?
Yeah, I know. I know it will help.
We're hoping that we'll discover not just a characterization of this disorder and what it looks like, but that we'll also discover distinct subtypes of autism. And if we can discover that there are 3 or even 10 different kinds of autism, that will be major progress. Autism is an incredibly complex disorder that just isn't understood at all. So this is a much harder problem than anything I've tried to do before. On the other hand, the stakes are higher. It really matters.
My name is Kai Goossens, and I'm an assistant professor at the McGovern Institute. As I transitioned from a graduate student to a postdoc, 2 members of my immediate family became severely mentally ill. As I was starting to plan my experiments, I was interested in the interaction between chronic stress and fear. Both of the members of my family who had mental illness, when they experienced major life stressors, it made their illnesses so much worse.
Kai is someone who wants to find a whole new biological approach to treating depression and anxiety disorders.
For a long time, we thought that we knew how chronic stress impacted the brain, that neurons actually shrink in response to chronic stress. But it turns out that that's not the whole story. When you chronically stress an animal, neurons in a part of the brain that is especially important for generating and expressing fear actually grow. The part of the brain that shows growth is called the amygdala. It's especially important in generating and expressing fear. We've identified now a number of genes that are turned on only in the amygdala following chronic stress. That is, chronic stress doesn't cause these genes to be turned on in any other part of the brain.
Linking those pieces together, linking from a gene to thoughts and emotions, I mean, this is an enormous undertaking.
Once you've identified genes in a particular brain region that you think are involved in something, you need to have a way to manipulate those genes and see which of those actually lead to changes in behavior. So I'm very hopeful that what we do in our lab will eventually result in better treatments to block the effects of chronic stress on mental illness.
Hello, I'm Ann Graybeal. Hi. How are you? When I was in school, The predominant thought was that a scientist would do the best science if he never thought about the possible application of his science. My attitude is now completely changed. I just want to do something that really is going to fundamentally help either patients or normal people in the way they lead their lives.
Anne Graybiel is interested in how the brain learns through experience. She's somebody who embodies the molecule-to-mind approach of the McGovern Institute.
We work on structures in the brain called the basal ganglia, which are right underneath the cortex on the outside. A number of years ago in our lab, we found that the chemicals in these basal ganglia are not evenly distributed, but instead they're concentrated differently in two main tissue types.
Anne's one of the world's experts in the neurotransmitter dopamine. As many people know, dopamine is what you lose in Parkinson's disease.
Great, Anne. Hi, come on in. Nice
We
to see you.
recently had
Nice
the
to see
honor
you.
of meeting an
Stanley.
MIT graduate, Mr. Sidney, and
Ken,
his
and you remember Anne,
daughter, also an MIT
Awards.
graduate.
Dad, because you've had the disease now for about 20 years, probably, right? About that long?
Mr. Sidney, who has severe Parkinson's disease, actually took the time to show the students and postdocs and staff members in our lab what it was like.
Hold your hands out. Do you have any tremor right now?
Mr. Sidney is being treated with a therapy called deep brain stimulation, or DBS for short. And in this technique, wires, electrodes, are put deep into the basal ganglia, and they apply a very small current. And the point of that current is to disrupt the abnormal activity that's going on at this little point. A wire leads off that electrode down to a permanently implanted control device, just like a pacemaker.
It's basically, if you can see the outline, it's like right that big.
I see.
Yeah, so that's about as large as they are. And they don't, if you want to feel it, he's going to show you. You can touch it.
Oh.
So it's just right under the skin.
The DBS in your case was done to help the motor symptoms, clearly.
Correct. Just feeling unable to make your body move and being trapped by your body or trapped and unable to calm your hand down was very, very uncomfortable for you.
DBS therapy has clearly helped Mr. Sidney's quality of life, but everyone in the field knows that we're going to have to do even better. In our lab, we're trying to do that by understanding how the basal ganglia work normally and what goes wrong with them in disorders such as Parkinson's disease.
That research that you're doing is going to affect countless lives and really gives us a lot of hope that, that his life will be improved and many others as well.
My name is Chris Moore. I'm an associate professor in the McGovern Institute. When I was 7, I started writing a book on the brain, but decided I should delay that to junior high school so I could finish it when I had more knowledge. The key to brain dynamics is understanding how your brain changes its organization on a millisecond to second timescale. One of the key observations that came out starting in the late 1920s is that often there's a rhythm to how your brain functions. Oscillations that happen, say, 40 times a second, and that's called the gamma rhythm. If you record from people's brains while they're performing tasks that ask them to shift their attention around, to listen to one thing as opposed to another, to pay attention to one part of the room as opposed to another, this 40 hertz oscillation shifts as your attention shifts.
The question is, what's the neural circuitry that gives rise to these rhythms, and what do these rhythms do?
Somehow, if we understand this, it might be a key to understanding, for example, diseases where the 40 hertz oscillations go bad. One of those diseases, for example, is schizophrenia. What's really been lacking is a way of turning on and turning off oscillations at will. We need a way of selectively driving one kind of oscillation or another in one brain area or another. And that kind of control has been lacking until now. There's a really remarkable new technology which allows cells to become optically sensitive, to be sensitive to light, that was co-developed by someone else here at the McGovern Institute, Ed Boyden.
We've been using optical control, or optogenetics as it's sometimes called, to understand what the causal role of a neuron is in behavior, pathology, and cognition. What I'm going to show you here is actually one of the world's first devices, which is now widely used in hundreds of labs around the world, that allows us to beam light down an optical fiber from a laser setup. This laser setup allows us to deliver blue light or yellow light down this fiber so we can turn neurons in the brain on or off. But neurons in the brain aren't naturally sensitive to light. We have to make them sensitive to light through our neuroengineering efforts.
So the way that we can make cells selective to light stimulation is by literally transferring genes to specific cells in specific brain areas.
And when light shines on these, it can actually activate electrically or silence electrically those very cells.
We're incredibly fortunate to have Ed Boyden here at the McGovern Institute at MIT. He continues to develop just amazing new optogenetic tools. This is a technology that people hadn't even speculated on 5 or 6 years ago.
Having obtained optical control over specific cell types in the brain, we can now ask, can we induce specific brain oscillations by driving that cell type?
We think that these gamma rhythms are involved in many of the brain's operations, and as we understand them more, we're going to understand a lot more about how they may go awry in a whole variety of mental disorders.
MIT has the absolutely perfect environment for developing the new techniques that allow us to ask the new questions, that allow us to open up new paths in neuroscience.
The McGovern Institute, for me, is a phenomenal addition to our scientific work and to the pleasantness of doing that work. It sounds so corny, but I'm extremely interested in the promise of neuroscience for society.
We can do absolutely anything here. There are no excuses. Any kind of expertise we need is right here in this building, right around us. I never end up in a situation where I think, oh, I really want to work on this problem, but I have no idea how and I don't know who to ask. I always know who to ask. They're right here. They're a couple doors down the hall.
If you want to combine techniques, the way to do it isn't to work by yourself. I think the McGovern Institute is one of the most collaborative environments that I have ever worked in.
I can't think of anything else comparable in the world that would allow radically new ideas from left field to get out there and you know, in one short year have huge impact on the world.
Once you understand the science of the brain, you can't think about the mind in the same way.
I've always viewed MIT as the cauldron of ideas, the place that we go to for new ideas that are going to really impact society. So this is where the government came to MIT for the development of radar that helped us win World War II. The government came to MIT for the technology that helped put us on the moon. And now, basically, we're coming to MIT and saying, where are the new ideas and the new technology that are going to give us these new treatments for brain disorders?
The curve of progress is virtually going up vertically. Things are happening so quickly.
The thing I love about being involved with the McGovern Institute is because I believe we give people hope. When there's hope and not defeat or despair on people's mind, progress will prevail and solutions will be found and neurodegenerative diseases will be cured. I just know that.